So now we've got an overview of the anatomy of the heart. We've looked a little bit at some of the diseases that can impact the heart. I want to now really start to focus physiologically on the heart. In other words, we're going to start looking at its function. We're going to start out by looking at the cellular level and how they function, and then look at more the coordination of heart contraction. So you'll remember from Bio 201, when you looked at histology, that there again are three types of muscle cells, one of which are cardiac cells. Cardiac cells are a type of muscle cell that are uninucleated. They have those intercalated discs. They're so striated, and they perform contraction, obviously, as all muscle tissue does. Now, one unique thing about these myocytes, these cardiac myocytes, is that not all cardiac myocytes contract. There is a small subgroup of cardiac myocytes which are called myogenic cells. Genic means to generate, to create, to bring forth. And these myogenic cells, their job is not to contract, rather their job is to generate a action potential. And it's called actually a pacemaker potential, because we're talking about the heart, and these are cells that help to, or that are responsible for, actually, the development of contraction in the heart muscle. They are what stimulate the other myocytes in the heart to contract. And here's an important point to know. The heart can beat, it can contract without any input at all from the nervous system, because of the fact that we have these myogenic cells that by themselves generate pacemaker potentials that signal all the rest of the muscle cells to contract. All that the nervous system does, and we do have innervation or connection to the nervous system, between the nervous system and the heart. All the nervous system is doing in that point is either telling the myogenic cells to speed up so the heart beats faster, or they're telling the myogenic cells to slow down so the heart beats slower. But they don't actually tell the heart to beat. The heart does that all on its own. You can disconnect all of the nerves to the heart, leading to the heart, and the heart will still beat because of these myogenic cells. The majority of the myogenic cells are located in a region of the right ventricle called the sinoatrial node, or the SA node for short. And I'm circling it right here for you so you can see. The SA node is also called the pacemaker location of the cell of the heart. And again, this is where we have the majority of the myogenic cells, and they generate a pacemaker potential about roughly 100 times per minute, which means that they, if they were left to their own devices, would cause the heart to beat about 100 times or beats per minute. And this is called the sinus rhythm, because it's derived from the sinoatrial node. Now, when the myogenic cells generate a pacemaker potential, and by the way, in a couple slides, I will show you how they do that. When they generate that pacemaker potential spreads out across the right atrium. It passes through the interatrial septum and over to the left atrium at the same time. It also hits a structure located in the sidewall of the atrium called the atrioventricular node. So those are all the places where the pacemaker potential spreads: right atrium, left atrium, atrioventricular node. As that pacemaker potential spreads to the right and left atria, they contract. As that signal reaches the atrioventricular node, it actually slows down for just a minute. The atrioventricular node is there as a connection point to some other structures that will take that potential down through the interventricular septum and to the ventricles. The atrioventricular node, as I said, causes a slowdown of the signal. And that slowdown of the signal is really important, because if I transmitted that signal simultaneously at the exact same moment that it traveled to the atria, that would give the ventricles the signal to contract at the exact same moment that the atria are contracting. And if you imagine, if I contracted all four chambers of the heart at exactly the same time, there would be no open place or open chamber into which I could dump the blood. When the atria contract, they are contracting to dump blood into the ventricles. If the ventricles were contract at the same moment as the atria wouldn't be able to squeeze the blood anywhere. So it's really important that we allow the atria to contract first and delay for just a few milliseconds the contraction of the ventricles so that it happens after the blood has been squeezed from the atria into the ventricles. So the atrioventricular node is what helps to delay the signal just briefly. And it does it in a really simplified manner like this. I'll give you a good analogy. If you've ever been in a highway construction zone where maybe you had, 4 or 5 lanes of traffic on a freeway, and because of a construction, it all gets funneled down to one single lane of traffic. You know that at the point before the merger, all the traffic slows down a whole bunch, right? The flow of traffic slows tremendously. Well, the same thing happens in the atrioventricular node. We have lots of points of entry for that pacemaker potential to come into the atrioventricular node, but very few exit points. And so we have lots of entry, just like we have lots of lanes of traffic coming in, but only 1 or 2 exit points going out. And so the signal slows down just a little bit, just like traffic slows down. The atrioventricular node, by the way, you'll see in some older textbooks, is called the bundle of His. That's its old name. I want to take some of this stuff I've drawn off the heart here so I can get to some of the structures that are further down the pathway from the AV node. So again, here's my AV node, and the AV node connects up with the AV bundle branches, I should say. So here are my bundle branches. You'll notice that there is a left bundle branch and there's a right bundle branch. And these left and right bundle branches then split off and they go down to the apex. Remember, that's the pointy part of the heart here. And then they turn and start traveling up the sides of the ventricular walls here. And when they turn and start spreading out along the ventricular walls and moving upward, then we call them Purkinje fibers, and I'll show that on the next slide. Before I do that, though, I want to mention that the atrioventricular node has its own internal rhythm because there are some myogenic cells that are also located in the atrioventricular node. If they were left to their own devices and they had no input from the SA node, they would beat at a rhythm of about 40 to 50 beats per minute. This is called nodal rhythm. Sometimes people can have a condition wherein either their SA node becomes damaged or the connection between the SA node and the atrioventricular node becomes damaged. Either way, what happens is the SA node is now out of the picture. And if that happens, the atrioventricular node will take over with the task of generating a pacemaker potential for the heart. If that happens, we have what's called total heart block. So in other words, in total heart block, if something happens and the signal can't get past the AV node, let's say the AV node itself gets damaged, then there's no way to pass that signal downstream to the ventricles. That's total heart block. Now, the only amount of delay that the AV node produces is really small. If you're interested, it's only about 100 millisecond delay, but it's just enough to let the atria contract before the ventricles contract so that blood moves forward from the atria to the ventricles and then out of the heart to either the pulmonic or systemic circuits. Now, I said next to the atrioventricular node, the next thing we have is the AV bundle. That's this structure right here that leads from the AV node. So the AV bundle then splits off, as I said, into the left and right bundle branches. And those bundle branches wrap up around the ventricles and they spread out into a network of smaller conducting pathways called the Purkinje fibers. And these structures altogether help to spread out the signal to contract along all of the ventricular surface simultaneously so that we get a good coordinated contraction of the ventricles exactly when we need them to contract. Sometimes things can go wrong, and we can get rogue cells, rogue myogenic cells that start generating their own pacemaker potential without being part of this circuitry. Or they may be part of the circuitry, but they just start doing their own thing. That's called an ectopic focus. So this can happen sometimes in patients as they get older, and obviously if you've got a rogue collection of cells generating their own pacemaker potential, that's going to interfere with the contraction, the coordination of contraction in other parts of the heart. And so we're going to have combating signals for heart contraction, and the overall outcome is that the heart does not beat in a coordinated fashion as it should. So as promised, I am going to talk now about how it is that those modified myocytes, those myogenic cells, how it is they go about generating this pacemaker potential, this continuous signal to the rest of the heart cells to contract. Now there's a couple things about myogenic cells I need to tell you. Normally, resting membrane potential for most cells in the body, you learned, is about 70 millivolts. And again, that's just an average. Different cells have different resting membrane potentials. But on average, you learned in 201 that it was about 70 millivolts. Myogenic cells, technically speaking, don't have a true resting potential, but we'll think of it as that they kind of do. And that pseudo resting membrane potential is around negative 60 millivolts. So here we are, right around here, negative 60 millivolts. Myogenic cells have a unique feature to them, which is that they have leaky sodium gates. These are sodium gates that are open all the time, and they allow for the leakage of sodium into the cell. Remember that if we look at a typical cell, it's high in potassium inside, low in potassium outside of the cell. By comparison, there's typically a high amount of sodium outside of every cell and a relatively low amount inside every cell. So if I have these sodium gates or these sodium channels that are open pretty much all the time, you're going to have a constant influx of sodium into the cell. Sodium is positively charged, and as it's positively charged, as more of it leaks in slowly, it's going to pull this cell up closer to threshold. Threshold for a myogenic cell is negative 40 millivolts. And that threshold of negative 40 millivolts, when it's hit, causes other types of voltage-sensitive gates to open. Okay, these are gates that are sensitive to changes in charge. When the charge gets to negative 40, these gates open, and these gates are what are called fast calcium gates. So they are specific to permitting the inflow of calcium, lots of calcium outside the cell, very little inside. The calcium rushes in very quickly. And again, because calcium is also positively charged, this is going to pull the cell up into the positive territory as more and more positive ions flow in. We get rapid depolarization of that myogenic cell. When we get up to around 0 or so millivolts, then those fast calcium gates close and fast potassium gates open. And as potassium rushes out of the cell becomes more negative. We get repolarization. And we get back down to pacemaker potential about negative 60 millivolts. This entire process happens about every 0.8 of a second. And again, this happens all without any input from the nervous system. This is just a process that occurs spontaneously because of the presence of these leaky sodium channels. Let's compare now the electrical activity of a myogenic cell that we just talked about with the actual contracting cells of the heart, the myocytes or the cardiac myocytes. These guys behave differently. Their job is not to generate a pacemaker potential. Their job, like other muscle cells, is to shorten or to contract. There are a few things about cardiac myocytes that make them different from, for example, skeletal muscle cells. One of those differences is that their resting membrane potential is about negative 90 millivolts. And once we get a pacemaker potential generated from those myogenic cells through the process I just talked about on the previous slide, this triggers the opening of voltage-sensitive sodium gates. So just to orient you here was we're looking here at the green line, which is the actual generation of the action potential. The red line here shows the actual contraction and relaxation of the cell in relationship to the action potential that travels across the surface of the cell. So here we are down at negative 90 millivolts. And we have these voltage-sensitive sodium gates, and they notice that there is a pacemaker potential that has reached the cell. And they are sensitive to that change in charge. They open up very rapidly, and lots of sodium rushes in very quickly, and you get this extremely steep depolarization. Again, these are rapidly opening sodium gates. Sodium rushes into the cell depolarizes, and gets up to around maybe positive 20 millivolts. And at this point, something unique happens that we don't see happen in skeletal muscle. What's unique that happens is a handful of calcium gates on the surface of the cell, on the plasma membrane, open up, and a little bit of calcium rushes into the myocyte. When that little bit of calcium rushes into the myocyte, it triggers the sarcoplasmic reticulum inside of the myocyte to release even more calcium. So a little tiny bit of calcium from outside of the cell rushes in. That little bit of calcium that rushes in then binds to gates on the sarcoplasmic reticulum and triggers the release of a whole lot of calcium inside of the cell. So now we have all this calcium inside of the cell, and what this does is it prolongs the amount of time that the cell is in the positive territory. The longer the cell is in that positive territory or depolarized, the longer we can sustain a muscular contraction. This is important because we don't want to just have a quick twitch contraction like we see in skeletal muscle. That wouldn't be a sufficient amount of time for contraction in order to eject all of the blood from the ventricles, for example. We need to have a contraction that is sustained for a few milliseconds so that we get enough time to squeeze all that blood out of the ventricle. So that's the purpose of having this calcium floating around in there for a few minutes, or not a few minutes, for a few milliseconds. And once that calcium has floated around for a little bit, then the sarcoplasmic reticulum will start its job of collecting the calcium back up. And at the same time, we start to get potassium moving out of the cell. And the combination of these two together helps the myocyte go back down into negative territory. In other words, it helps it repolarize back to its original resting membrane potential. These calcium gates are typically of the slow variety, we think, which is why calcium tends to linger in the cell for a relatively longer period of time than we might otherwise see, say, in skeletal muscle. And so because the calcium lingers inside the cell, we get a longer period of contraction. We can measure this electrical activity that's happening inside of a heart through the use of something called an electrocardiogram, or ECG. And an ECG is it's a piece of equipment that has some wires, or what are called leads, that lead off of the machine and connect on the patient. And they're put at various places on the patient. One goes on each wrist, for example. There's one that goes on the leg, and then there's a series of leads that go across the chest region. Collectively, these all are points of monitoring for that machine, where it can monitor electrical charges, and these electrical charges are representative of both the pacemaker potential that's being generated and the actual contraction and relaxation of the heart muscle. And I want to take you through what a basic ECG wave looks like. If we were to, in other words, draw or plot the changes in charge that are occurring over the course of, say, a 1 second time period or one complete contraction and relaxation of the heart, here's what it would look like. Here's 0 millivolts, here's positive 1 and negative 1 millivolts. We start out at 0 millivolts on the leads, And the first thing that happens is we get a pacemaker potential generated, a process of depolarization, and that's represented by this thing called the P wave. And this P wave represents a depolarization of the atria. So the atria depolarize, and then we have a segment here from P to what's called Q, the P to Q segment, or the P to Q line. And the P to Q line represents the actual atrial contraction. Notice that this is a flat line. We're not having any change of charge going on right now. We're just having contraction occurring. So first we have the P wave, which is depolarization of the atria. Then the atria contract. That's the PQ line. Now the next thing that would happen is the atria relax, and they do that. They happen to be doing that at exactly the same time, though, that the ventricles are depolarizing. And because the ventricles are really much larger structures, there's more cells in the ventricles than in the atria, the ventricular depolarization event sort of masks or covers over or hides the repolarization of the atria. So the QRS complex here, which is representative of ventricular depolarization, is happening at the same time that atrial repolarization is occurring. So all we really see is the ventricular depolarization. Just remember that atrial repolarization is also occurring at the same time. After we get the ventricles depolarizing, the next thing that needs to happen is the ventricles need to contract. And that contraction happens between the S and T line, or the S and T segment here, between S and T. That's ventricular contraction. Again, it's a flat line. We don't have any changes of charge occurring. We've just got shortening of the muscles, of the muscle cells. And then the last thing we have is ventricular repolarization, and that's represented by this thing called the T wave, this little bump right here. That's ventricular repolarization. After the ventricles repolarize, the last thing they do is they relax. And that's this flat line over here. All right. Now, two terms I want to just toss out here if you didn't know them already. Another word for contraction is systole. Another term for relaxation of heart muscle tissue is diastole. You hear those terms used quite a bit. So contraction, systole, relaxation, diastole. So let's take a look at this one more time. I'll do it for you again because it never hurts to see something more than once. So again, here we are. We're in the atria. The SA node sends out a signal. The myocytes depolarize, and as they depolarize, we create a P wave on our ECG. The atria then contract, and that is my P to Q line, or P to Q segment. Then the atria relax at the same time that the ventricles depolarize, and because the ventricles depolarize with a greater noise, because there's more cells, there's more electrical charge going on, they kind of cover over or mask over the relaxation of the atria. We get this QRS complex that represents ventricular depolarization, and then the ventricles undergo contraction, or systole. That systole is represented by the S to T line, or the S to T segment. And then the last thing that happens is in this T wave here, we get repolarization of the ventricles. That's the T wave, and then after that they relax, and that's the post-T line. We'll go over this some more in class so you get some more practice with it too. Based on the information we gather from an ECG, a cardiologist can tell a lot about her patient. She can determine if that patient maybe has recently experienced a heart attack. She can tell whether that person might be having some sort of an ectopic focus somewhere in the heart. She can tell if there's total heart block. She can tell if there might be some sort of fibrillation occurring. She can tell all kinds of things about the patient based on that ECG, based on how big the P, the T, and the QRS waves are, based on their timing, based on their absence, all of those things. So let me take you through just a couple of examples of this. Here's a normal sinus rhythm. This is what a normal person has. The P wave followed by the QRS complex followed by the T wave. In this patient here, you can see that there's no P wave before the QRS complex. And what that tells you is, remember, the P wave signifies atrial depolarization. So that's telling us that the atria, the SA node in the atrium is not active the way it's supposed to be. And as a result, the AV node has had to take over instead, and we have a nodal rhythm rather than a sinus rhythm controlling the heart. If we have no ability to convey the signal from the SA node to the bundle branches and the Purkinje fibers, etc. That was, remember what we said, was heart block, and it usually happens if the AV node has been damaged. Then we get something that looks like this, where you see P waves occurring, but no QRS complex and T wave afterwards. That tells you that the ventricles are not depolarizing, therefore they are not contracting. If I have a really large P wave, and I don't have a picture of this, sorry, but a really large P wave usually indicates that you have mitral valve stenosis, or that narrowing of the left atrioventricular valve. If I have a really big Q part of the QRS complex, that's usually indicative of a myocardial infarction. If the R part of the QRS complex, that's the high peak, if that R wave is really large, then that usually is an indication that the ventricles have enlarged through a process called ventricular hypertrophy. That means the individual cells of the ventricles have gotten larger. Sometimes this can happen normally in really high-performance athletes. Sometimes it happens because of certain heart conditions like hypertension. Sometimes you can get what are called arrhythmias. This means that the heart is not beating in a coordinated fashion. That can happen because of things like the AV block I talked about here, the heart block, or it can happen because of fibrillation. And fibrillation means a loss of coordination of contraction. And it looks like this. If you were to look at, for example, ventricular fibrillation, you have absolutely no discernible pattern. There's no predictable QRS complex, no predictable P or T waves. It's just kind of a mess of electrical signals with no pattern to them. That's fibrillation. You can have the atria undergo fibrillation. You can have the ventricles undergo fibrillation. Obviously, because the ventricles are what actually force the blood either to the pulmonary or systemic circuits. If the ventricles fibrillate, that's a really serious life-threatening condition. Whereas if the atria fibrillate, if they develop an arrhythmia, you can still fill the ventricles for the most part because of a process called passive filling that I'm going to get to on the next portion of this lecture on the cardiac cycle. So if the atria fibrillate, it's usually not a life-threatening condition. If the ventricles fibrillate, you have a very serious medical condition that has to be treated immediately. Usually they do that by shocking the patient with what's called a defibrillator to remove that fibrillation and establish a coordinated contraction cycle again.